All right, so chapter 15 is going to be our final chapter in the genetics unit, and we're going to talk about biotechnology. So basically, how did we discover the genes that we know about today, both in things like bacteria and animals and humans and plants, and also how do we do experiments with those genes. So what kind of technology do we need to do those experiments? And then we'll talk a little bit about how can those genes be changed? How can those technologies be used for things? So first of all, I want to talk about probably the biggest genetic project that's ever happened is the Human Genome Project. And the Human Genome Project began in 1990 and it was completed in 2003. So it's pretty crazy. This project, the start of it anyway, is about 30 years old. So 30 years ago is when we first started looking into how do we map the functions and locations of genes on human chromosomes. So the first gene that we sequenced was very expensive, right? So the very first time we did this, it cost millions of dollars, and it's not something that everybody could do. Now, today, you can send a way to have your genome sequenced, and it'll probably only cost you about 20 bucks. It's not super expensive. And that would be what it would cost you. What it costs a company to sequence your genes is not very much at all. So this process, as we've gotten better at it, has gotten much cheaper. But the first time we did this and developing all the technology was difficult. The major purpose of that project was to try to find out why or how, like what influence genes had on diseases. And we thought that it would give us a lot of information for curing diseases, and it certainly did. But it wasn't quite exactly like people thought it would be. So people thought once we know all the genes, we'll be able to map the genes, we'll be able to find problems with the genes, and then we should fairly immediately be able to cure, a lot of these things. And it turned out that there were quite a few complicating factors. When we sequenced the human genome, we thought there were going to be many more genes than there actually ended up being. We ended up finding about 20,500 genes. People had estimated that there would be more than that. It turns out that the human genome sequence is about 3 billion base pairs, so it is quite large. But it doesn't have as many genes as we thought it would have for the size in base pairs. So before we could get to the human genome and sequencing that, we had to sequence some things that were smaller and simpler. So we didn't begin with humans as the first organism whose genome was sequenced. We did a variety of other things. So E. coli had its gene sequenced, yeast was a common one, and then the roundworm, known as C. elegans, also had its gene studied and mapped. All three of these are organisms with much smaller genomes. So it made them a little bit easier to sequence and a little, certainly cheaper to sequence. And then E. coli is one of the first ones because it has the circular DNA. Okay, in comparison to humans, E. coli is about 4 billion base pairs, and it has only 4,000 genes. So it's a much easier genome to sequence. Sequencing each one of these genes helps us to understand a little bit more about sequencing itself, and also helped us to understand the similarities and differences between organisms with different genotypes or genomes. Just a note on similarity as far as base pairs go. If you were to just basically count the number of base pairs and then identify base pairs that were the same in different organisms, humans are about 98.5% similar to all other humans. If you have identical twins, they're about 99.9% similar to each other, so almost 100% similar. If you were to compare your genome with that of a chimpanzee, it would be about 98% similar. If you were to compare your genome with a banana, you'd be about 50% similar to a banana. So similarity between genomes certainly does tell us how related organisms are, but just because an organism has quite a few similarities in common with another organism does not necessarily mean that structurally they're going to appear the same, right? I don't think you could say that structurally or behaviorally you are 50% the same as a banana, but your genes are 50%. They would have in common, okay? So when we're talking about different gene technologies, we're going to talk about a few things. So first of all, if we wanted to do anything with gene technologies, we would have to access your DNA. I don't really have a slide in here on how we access DNA, but I did just want to go through and explain some of that. So the first way that we would access DNA would be to do something to break the cell open and release the DNA. Usually the word for breaking the cell open would be to lyse the cell, and we would call this extracting DNA. So to get DNA to work with out of cells, we often have to open those cells up and extract that DNA in some way. Now I'm going to go through and I'm going to talk about several processes that would be used to either alter genes or to detect those genes and then use them for different purposes. Before I get into that, I'm going to go ahead and pause this lecture, and then we'll switch to a second one so you guys can get caught up on notes or take some more notes if you need to.